Right now, as you're listening to this, there's something ancient living inside your cells, something that isn't quite you. Fragments of DNA from a human species that vanished from the Earth 30,000 years ago, but never really disappeared. They're still here, still alive, still part of you. Think about that for a second. A species went extinct before the pyramids, before agriculture, before we even figured out how to make pottery, and yet they didn't completely die out. Because somewhere in your genome, in the twisted ladder of molecules that makes you who you are, there are pieces of them. Instructions written by a people who lived in a world so different from ours it might as well be another planet. If you're using this to fall asleep, that's exactly why I make these videos. So maybe subscribe and tell me where you are and what time it is. I love seeing the comments. Tokyo at sunrise, London at midnight, all listening to the same ancient story. This is the mystery of the Denisovans, a human species we barely knew existed until 2010, discovered not through massive fossil beds or sprawling archaeological sites, but through a single tiny finger bone from a cave in Siberia. One bone. That's all it took to rewrite the entire story of human evolution. Here's what makes this so mind-blowing. We don't know what they looked like. We have almost no fossils. We don't know how they lived, what languages they spoke, whether they made art or music. We're not even entirely sure where they lived, though the evidence keeps turning up in the strangest places. And yet, we know they were real, because billions of people alive today carry their DNA. How does a species go extinct, but remain alive inside us? How do you leave almost no trace in the ground but an indelible mark in human genetics? How do you vanish from the earth but survive in the bodies of people climbing mountains in Tibet, diving in the waters off Indonesia, walking the streets of Sydney and Shanghai? If you've been enjoying these deep dives into our ancient past, we've actually put together something special. A whole book exploring prehistoric secrets and the hidden history written in our genes, available over at sleepytimehistory.com. But we're just getting started here. Tonight, we're going to follow the trail of evidence from a remote cave at the edge of Siberia to the cutting edge of genetic science. We'll meet the scientists who discovered an entirely new human species from DNA alone. We'll explore how these mysterious people lived alongside Neanderthals and our own ancestors in a world shared by multiple human species. We'll uncover the love stories, because yes, our ancestors didn't just meet the Denisovans, they had children with them. And we'll discover why some of their genes turned out to be the key to human survival in the most extreme environments on Earth. The Denisovans are ghosts, but ghosts that left something behind, something that changed us forever. The Altai Mountains rise from the heart of Central Asia like the spine of the world. This is where Russia bleeds into Mongolia, where Kazakhstan meets China, where ancient migration routes crossed paths for hundreds of thousands of years. It's remote, brutally cold in winter, hauntingly beautiful in summer when the valleys fill with wildflowers and the rivers run clear with snowmelt. And it's exactly the kind of place you'd expect to find nothing important. Except there's a cave, Denisova Cave, named after Denis, an 18th century hermit who supposedly lived there, seeking isolation from the world. He probably had no idea he'd chosen one of the most important archaeological sites in human history, a place that had sheltered human beings on and off for over 300,000 years. Think about that timeline, 300,000 years of continuous or near-continuous occupation. The cave was old when Neanderthals first walked into it. It was ancient when modern humans arrived. Generation after generation, millennium after millennium, different human species came to this place, lived in it, died in it, left their bones and tools and fires buried in the sediment. By the time Russian archaeologists started systematic excavations in the 1970s, the cave floor had become a layer cake of human history. Each stratum told a story. stone tools from different traditions, evidence of fire use stretching back hundreds of thousands of years, bones from animals that had been hunted, butchered, consumed, and mixed in with all of it, fragments of the hunters themselves. The cave wasn't huge, not some massive cavern system, just a modest limestone hollow carved by an ancient river, maybe 30 meters wide at its mouth, extending back into the mountainside in a series of connected chambers. But its position was perfect, sheltered from the worst weather, close to fresh water, overlooking a valley rich with game, the kind of place any sensible human would choose as a base camp. For decades the excavations proceeded slowly, carefully, revealing layer after layer of occupation. The archaeologists found Neanderthal remains. They found evidence of modern humans. They documented the transition between different stone tool technologies, the changing climate recorded in pollen and sediment, the rise and fall of different animal populations. It was important work, solid archaeology, but not revolutionary. Then in 2008, they found something small, unremarkable at first glance. A fragment of bone from what appeared to be a young girl's finger. Technically, it's called a distal phalanx, the last bone in a finger, the part closest to the fingernail. This one was tiny, maybe 1.5 centimeters long, probably from a child somewhere between 5 and 7 years old. The bone came from layer 11, deep in the cave's stratigraphy, from sediments dating to somewhere between 30,000 and 50,000 years ago. Right in the middle of the last ice age, when Neanderthals still roamed Europe and modern humans were just beginning their expansion across Eurasia. At the time, nobody thought it was particularly special. Old human bones turn up in archaeological sites all the time. The Russian team catalogued it, photographed it, and did what researchers often do with interesting finds that might yield more information. They sent a sample to specialists. The bone fragment made its way to Leipzig, Germany, to the Max Planck Institute for Evolutionary Anthropology, to a laboratory run by a Swedish geneticist named Svante Pääbo, who had spent decades developing techniques to extract and sequence DNA from ancient remains. Pääbo's lab had already made history. They'd been the first to successfully sequence Neanderthal DNA, proving that genetic material could survive tens of thousands of years under the right conditions. They'd shown that modern humans and Neanderthals had interbred. They'd revolutionized our understanding of human evolution. But they had no idea what was hiding in that finger bone. The initial extraction seemed routine. Ancient DNA is always degraded, broken into tiny fragments by time and chemistry and the simple process of decay. You don't get long, intact sequences. You get molecular scraps that have to be painstakingly pieced together, like assembling a jigsaw puzzle, where 99% of the pieces are missing and the remaining 1% are broken. But there was DNA. Good quality DNA, actually, preserved by the cool, stable conditions in the cave. The team began sequencing it, comparing it to the reference genomes they'd already assembled from Neanderthals and modern humans. And that's when everything changed. The DNA didn't match. It wasn't modern human. But it also wasn't Neanderthal. The genetic signatures were wrong. The mutation patterns were different. This bone fragment, this tiny piece of a child's finger found in a remote Siberian cave, belonged to something else entirely. A human species nobody knew existed. The team ran the analysis again, then again. They checked for contamination, for errors in sequencing, for any possible explanation that would make the data fit into the known framework of human evolution. Nothing worked. The conclusion was inescapable. They'd found a new type of human, one that had been living in Asia while Neanderthals dominated Europe, and modern humans were still mostly confined to Africa, one that had left almost no fossil trace, one that had been completely unknown to science until a child's finger bone revealed its existence through DNA alone. They called them Denisovans, after the cave, after the hermit who'd never known he was living among the remains of humanity's forgotten cousins. And the discovery was just beginning. Here's what you need to understand about DNA. Under normal conditions, it doesn't last. The moment an organism dies, the molecular machinery that kept everything running starts breaking down. Enzymes that were carefully regulated suddenly run wild, cutting and chopping genetic material into smaller and smaller pieces. Water seeps in, causing chemical reactions that destroy base pairs. Bacteria move in and consume whatever's left. Within days, weeks at most, DNA degrades into molecular noise. This is why ancient DNA research seemed impossible for most of the 20th century. Scientists could extract and sequence DNA from living things, even from recently deceased specimens. But anything older than a few decades? The genetic material was just gone, degraded beyond recovery, except in very specific circumstances. Cold helps. Dryness helps even more. Lack of oxygen is crucial, and stable pH conditions can preserve molecular structures that would otherwise fall apart. Put all these factors together, and you get places like Denisova Cave, limestone formations with stable temperatures hovering just above freezing, low humidity, sediments that protected buried remains from oxygen exposure, the kind of environment where DNA doesn't decay normally, where genetic material can survive not for years or decades, but for tens of thousands of years. Even then, what survives isn't pristine. Ancient DNA comes out in fragments, tiny molecular pieces, often just 50 to 100 base pairs long, when functional DNA sequences can be millions of base pairs. It's like trying to reconstruct a library from scraps of paper that have been torn up, scattered, exposed to the elements for millennia, then partially digested by bacteria. For most of the late 20th century, this seemed like an impossible problem. Researchers could occasionally extract ancient DNA from relatively recent specimens, mammoths frozen in permafrost, Egyptian mummies preserved by desiccation. But the sequences were fragmentary, contaminated, difficult to verify. Many early claims of ancient DNA recovery turned out to be contamination from modern sources. Then came Svante Pääbo, a Swedish biochemist who became obsessed with the idea that genetic material from extinct humans might still be recoverable, not just theoretically recoverable, but actually accessible with the right techniques and enough patience. Through the 1990s and early 2000s, Pääbo's team at the Max Planck Institute developed increasingly sophisticated methods for working with degraded genetic material. They figured out how to isolate ancient DNA from the bacterial contamination that fills most archaeological samples. They developed techniques for amplifying tiny fragments without introducing modern contamination. They created statistical methods for distinguishing authentic, ancient sequences from background noise. Most importantly, they learned where to look. Bones from cold, dry caves. Teeth, which preserve DNA better than other skeletal elements. Samples from sites with stable environmental conditions. Places like Denisova Cave, where the molecular clock had effectively stopped tens of thousands of years ago. By 2008, Pääbo's team had already achieved the impossible once. They'd successfully sequenced DNA from Neanderthal bones, proving that genetic material could survive from specimens over 40,000 years old. More than that, they'd assembled enough Neanderthal genome to compare it with modern human DNA and show something revolutionary. We'd interbred. Modern humans and Neanderthals had met, mated, and produced fertile offspring. Most people of European and Asian descent carry 1 to 2% Neanderthal DNA. This was the context when that fingerbone arrived from Siberia. The team had proven ancient human DNA recovery was possible. They had reference genomes from both Neanderthals and modern humans. They had the technical expertise and statistical tools to analyze fragmentary genetic sequences. They were ready, but nothing prepared them for what they found. The first analysis focused on mitochondrial DNA. This was standard practice. Mitochondrial DNA comes from the cellular powerhouses inherited exclusively through the maternal line, and it's present in hundreds of copies per cell compared to just 2 copies of nuclear DNA. This redundancy means mitochondrial sequences are much more likely to survive degradation. You can extract usable mitochondrial DNA from samples where nuclear DNA is too degraded to sequence. The team extracted mitochondrial DNA from the finger bone, sequenced it, and compared it to their reference databases. Modern human mitochondrial DNA differs from Neanderthal mitochondrial DNA by about 200 mutations accumulated over roughly 500,000 years of separate evolution. The Denisova finger bone was twice as different. About 400 mutations separated it from modern humans. Roughly 350 mutations separated it from Neanderthals. This suggested a population that had diverged from our lineage even earlier than Neanderthals had, then evolved independently for hundreds of thousands of years in complete isolation from both groups. The implications were staggering. Either they'd found the most divergent Neanderthal ever discovered, or they'd found something entirely new. Something that had been living in Asia while Neanderthals inhabited Europe, and modern humans evolved in Africa. A third human species that nobody knew existed. But mitochondrial DNA only tells part of the story. It represents a single genetic lineage passed down through mothers. To really understand what they'd found, the team needed the nuclear genome. The 3 billion base pairs of DNA contained in the cell nucleus that carries the complete genetic blueprint. This was much harder. Nuclear DNA is present in only 2 copies per cell, making it far less likely to survive. The fragments recovered from ancient samples are shorter, more degraded, more contaminated. Assembling a complete nuclear genome from 50,000-year-old bone fragments required processing billions of individual DNA sequences and computationally sorting them from modern contamination and bacterial DNA. It took months. The team extracted every possible fragment of genetic material from the fingerbone sample. They sequenced and resequenced, building up coverage 1 fragment at a time, using statistical methods to verify each sequence was authentic ancient DNA, rather than contamination. They compared every fragment to both modern human and Neanderthal reference genomes, looking for patterns of similarity and difference. Gradually a picture emerged. The nuclear genome confirmed what the mitochondrial DNA had suggested. This wasn't a weird Neanderthal. This was a distinct population that had separated from the modern human lineage around 800,000 years ago. Slightly before Neanderthals split off, they'd evolved independently across Asia for 750,000 years. They'd developed their own unique genetic signatures, their own adaptations, their own evolutionary trajectory, and then, at some point, they disappeared, leaving almost no fossil trace. No distinctive skull shapes that paleontologists could identify, no unique bone structures that would show up in archaeological surveys. Just DNA. Genetic sequences preserved in a child's finger bone from a Siberian cave, and, as it would turn out, scattered through the genomes of living people across Asia and Oceania. For the first time in history, scientists had identified a human species not from its anatomy, but from its genes. The Denisovans existed primarily as information, as sequences of base pairs that told a story of divergence, adaptation, and eventual extinction. They were a people known almost entirely through the molecular signatures they'd left behind. And those signatures were about to reveal something even more extraordinary. Here's the fundamental paradox that makes Denisovan research so strange. We know more about their genetics than we know about the genetics of most living species. We can trace their ancestry back nearly 1 million years, identify specific gene variants they carried, determine when and where they interbred with other human populations. We can sequence their entire genome with stunning precision. But if you asked anyone on Earth what a Denisovan actually looked like, nobody could tell you with any confidence. Because we have almost nothing to look at. A finger bone from a teenage girl, 3 teeth, a jawbone, that's it. That's the entire physical record of a human species that lived for hundreds of thousands of years across half of Asia. A people who developed technologies, built cultures, adapted to environments from tropical rainforests to the highest plateaus on Earth. And we're trying to reconstruct them from scraps that would barely fill a shoebox. Start with those teeth. They're weird, really weird. Enormous compared to modern human teeth. Even bigger than Neanderthal molars. The crowns are massive, the roots robust and thick. These teeth belong to people who were chewing something tough, regularly, and whose jaws were powerful enough to handle serious mechanical stress. The wear pattern suggests they weren't just eating soft fruits and cooked meat. They were processing food that required real bite force, maybe tough plant fibers, raw meat, materials that needed to be ground down before swallowing. The size suggests something else too. Dental dimensions correlate with overall body size across primates. Those massive molars probably belong to large-bodied individuals, not necessarily tall but robust, thick-boned, heavily muscled, built for power rather than gracility. Think Neanderthal proportions, but potentially even more rugged. Then there's the jawbone. The Xiahe mandible, named for the county in China where it was found, but the story of its discovery is almost absurd in its randomness. In the 1980s, a Buddhist monk was exploring the Baixia Karst cave on the Tibetan plateau, over 3,000 meters above sea level. He found what was clearly an ancient jawbone embedded in the cave wall, recognized it as something potentially significant, and donated it to a local monastery, where it sat for decades. Just sitting there, this incredibly rare specimen in a religious institution in one of the most remote regions on Earth. Nobody identified what it was until 2019. That's when researchers finally managed to extract protein sequences from the bone, ancient proteins that survive even longer than DNA in many conditions. The protein signatures matched Denisovan genetic patterns. This wasn't just any ancient human jaw. This was Denisovan, and it was found at 3,280 meters elevation in an environment that would challenge most modern humans without extensive acclimatization. The Denisovans weren't just living in Siberian caves. They'd colonized the roof of the world. The mandible itself is massive. Thick bone, large teeth, a robust structure that again suggests powerful jaw muscles and serious chewing capability. But there's something else interesting. It shows certain archaic features, characteristics that look more like much earlier human species. Homo erectus level primitive in some details. This suggests Denisovans retained ancestral traits that both modern humans and Neanderthals had lost. They were following their own evolutionary trajectory, keeping some ancient features while developing their own unique adaptations. But trying to picture a complete Denisovan from a finger bone, 3 teeth and a jawbone is nearly impossible. It's like trying to reconstruct what someone looked like from a photograph of their left thumb, a couple of toenails and their chin. You can make educated guesses, but you're really just speculating. That's where things get creative. In recent years, researchers have tried using DNA methylation patterns to reconstruct Denisovan anatomy. Methylation is how genes get turned on or off during development. By analyzing methylation patterns preserved in ancient DNA, you can theoretically predict which genes were active during growth, and from that, infer what physical features would have developed. These analyses suggest Denisovans might have had very broad faces, wider than either Neanderthals or modern humans. Their skulls were possibly quite wide as well, with prominent brow ridges and a projecting jaw. Some reconstructions picture them looking distinctly archaic, almost more Homo erectus-like than contemporary with modern humans. Others suggest features intermediate between different human species. But these are educated guesses built on statistical models based on fragmentary evidence. Nobody really knows. The reconstructions are hypotheses, not photographs. What about their culture, their technology, the lives they actually lived? Here the evidence gets even thinner. Denisova Cave contains layers where Denisovan DNA is present, sediments deposited during periods when they presumably occupied the site. These layers contain tools, but here's the problem. The tools look pretty much like tools made by Neanderthals and early modern humans in the same time periods. Stone points, scrapers, cutting implements. Sophisticated enough, but not distinctively different. Does this mean Denisovans lacked unique cultural traditions? Or does it mean stone tools represent convergent solutions to universal problems, and the real cultural differences were in organic materials that haven't survived? Did they have distinctive clothing styles, unique artistic traditions, different social structures? We have no idea. The archaeological record is silent. There's tentative evidence they created jewellery. Some bone ornaments and beads from Denisova Cave might be associated with Denisovan occupation layers, though the dating is uncertain. If confirmed, this would suggest symbolic thinking, aesthetic sense, possibly status display or ritual behaviour. But again, the evidence is fragmentary and contested. What we're left with is an entire human species that exists more as concept than reality. A people who thrived for hundreds of thousands of years, who adapted to environments from Siberia to the tropics, who interbred with both Neanderthals and modern humans in multiple locations over vast stretches of time. They were successful. They were widespread. They were human in every meaningful sense. But we can barely picture them. This is humbling in a way that's hard to articulate. An entire branch of humanity, as real as Neanderthals, as accomplished as any human population that's ever existed, and we're reconstructing them from genetic shadows and dental measurements. They had faces, voices, individual personalities, hopes and fears and stories. They looked up at the same stars we see, felt the same sun on their skin, loved their children and mourned their dead, and we don't even know what colour their eyes were. If Denisovans feel abstract and distant, Denisova Cave itself does not. Imagine a single place on Earth that watched the whole drama of our genus unfold. A cave overlooking a cold Siberian valley, layers of sediment stacked like the pages of a book, each one recording a different chapter of human presence. Not one species, not one culture, but many, coming and going, returning, disappearing, returning again. Denisova Cave is that kind of place. When researchers began dating the sediments and sequencing DNA from the dirt itself, they realized something astonishing. This wasn't just a Denisovan hideout. It was a timeshare. Over hundreds of thousands of years, 3 different kinds of humans used this cave as shelter. Neanderthals occupied it. Denisovans occupied it. Later, early modern humans came through as well. Their traces are stacked on top of each other, sometimes separated by tens of thousands of years, sometimes overlapping in ways that are still being sorted out. Same cave, same hearths, different species of humans sitting around the fire. In some layers, the DNA is almost purely Denisovan. In others, Neanderthal signatures dominate. Higher up you begin to see traces that match our own lineage. The stone tools don't always help you tell them apart. A point, a scraper, a blade made by a Denisovan hand might look almost identical to one made by a Neanderthal. The rocks won't tell you whose fingers held them. The DNA will. And then, buried in that layered history, researchers found something that changed everything. A broken piece of long bone, so small and unremarkable that it might easily have been overlooked. It didn't look like much. But when they sequenced its DNA, they realized they were looking at one of the most intimate pieces of evidence in all of human prehistory. The bone belonged to a teenage girl. She lived around 90,000 years ago. The genetic analysis showed something no one had ever seen before in ancient DNA. Half her genome looked Neanderthal, half Denisovan. Her mother was Neanderthal. Her father was Denisovan. She has a nickname now, Denny. Not a species name, not a Latin binomial, but a human nickname, because it's hard not to feel close to someone whose parents we can identify at the species level. Her genome doesn't show signs of distant mixing generations back. She's first generation, a direct hybrid. One parent from one human species, one parent from another, both of them carrying their own long evolutionary histories. Both of them descended from those bottleneck survivors almost 1 million years earlier. Their child, sitting somewhere near the mouth of that cave, is the proof that those histories were still intertwined. Denny's existence tells us that when Neanderthals and Denisovans met, they didn't only compete, at least sometimes they formed families, and if we're lucky, her bone fragment is not an exception. It's a glimpse of something that probably happened many times, in many places, over thousands of years. To really feel the shock of that, it helps to picture the world she lived in. Between about 50,000 and 100,000 years ago, Eurasia was not a continent dominated by a single kind of human. It was a mosaic, a patchwork quilt of different human species, each occupying their own regions, their own preferred environments, their own cultural landscapes. In Ice Age Europe and parts of Western Asia, Neanderthals had home field advantage. For hundreds of thousands of years they had refined the art of surviving cold seasonal climates. Short, powerful bodies, enormous strength, large brains, complex hunting strategies honed against mammoths and woolly rhinoceroses, deep knowledge of local landscapes. To the east, the Denisovans ranged across an astonishing variety of habitats. We catch glimpses of them in Siberia, on the Tibetan Plateau, and, through their DNA, in populations descended from people who lived in Southeast Asia and Oceania, from permafrost caves to tropical forests and island coasts. They seem to have been the great generalists of Eurasia. Comfortable in thin air on the roof of the world, but also present in rainforests and possibly along seaways. Farther south and west, in Africa and eventually spilling out into the Middle East and beyond, early Homo sapiens were developing their own set of strategies. More gracile bodies, flexible technologies, rapid cultural change, the ability to exploit a ridiculous range of resources, from shellfish to savanna game to forest plants. If you could look at a map of that time, not with political borders but with overlapping clouds of presence, you would see Europe shaded Neanderthal, much of mainland Asia touched by Denisovans, Africa lit with Homo sapiens, with tendrils of our species reaching into the Levant and occasionally farther. Those clouds wouldn't have been neat, separate circles. They would blur and overlap. Valleys where Neanderthal hunting ranges met Denisovan territories. Mountain passes where Denisovan high-altitude specialists occasionally descended and encountered lowland groups. Coastal corridors where early modern humans, pushing out of Africa and the Near East, ran into neighbours who'd been in Eurasia long before them. Each overlap zone was a potential contact zone, for trade, for conflict, for cautious watching from a distance, and sometimes for intimacy. For a long time, scientists assumed that if different human species coexisted, contact must have been rare and limited, separate branches on a tidy evolutionary tree, one branch replacing another, a neat succession. The genetics tell a different story. Modern people whose ancestry traces back to Europe, the Middle East and much of Asia carry Neanderthal DNA. People with ancestry from parts of East Asia, the Pacific and the Americas often carry distinct Denisovan DNA as well, These aren't tiny one-off traces. In some populations, several % of the genome clearly descends from these other humans. To leave that kind of signature, encounters couldn't have been vanishingly rare. They must have happened again and again, across thousands of years, in different regions, under different circumstances, sometimes perhaps in violence, sometimes in alliance, sometimes in more ordinary, complicated human ways that we will never fully reconstruct. Denisova Cave gives us one extraordinary data point: Denny, the first-generation hybrid. But when geneticists look at both Neanderthal and Denisovan genomes, they see hints of multiple episodes of mixing, stretching back hundreds of thousands of years. Neanderthals with bits of Denisovan DNA, Denisovans with fragments that look more like early modern humans, our own genomes with segments from both. It's not a straight tree, it's a braided river. Branches split, yes. Populations diverge. Neanderthals go one way, Denisovans another, Homo sapiens another. But then the channels rejoin for a while. Water from one branch flows into another, mixes, and then separates again. Generation after generation, the boundaries blur. The old picture of human evolution, one species turning into another in a clean stepwise progression, starts to look more like a comforting bedtime story than a description of reality. The truth, as far as we can tell, is messier and more interesting. For long stretches of time, there wasn't the human species. There were human species, plural, all of them intelligent, all of them capable, all of them shaped by the same ancient bottleneck, yet each adapting to its own ecological niche. Neanderthals specialized in cold, seasonal environments, carving out a powerful presence in Europe and Western Asia. Denisovans spread wide across Asia, from high plateaus to equatorial forests, possibly excelling in environments that demanded both physical robustness and physiological flexibility. Homo sapiens, initially confined to Africa, honed a different kind of flexibility, cultural and technological, before expanding into territories already claimed by others. They were not isolated experiments, each in its own sealed laboratory. They saw each other, they learned from each other, perhaps copied tools, perhaps stalked the same herds, perhaps told stories about the strange people beyond the next mountain range. And sometimes they had children together, whose descendants are still alive today. When you think about that, your own identity becomes a little more fluid. The borders between us and them start to dissolve. The Neanderthals are not just an extinct cousin. The Denisovans are not just a mysterious ghost in the cave sediments. They are threads in the tapestry that became you. The world they shared was harsher than ours in many ways, colder, more dangerous, full of predators and sudden shifts in climate, but also richer in one profound respect. It was a world where more than one kind of human walked the earth at the same time, a world where human was already a plural word. By the time our own branch of humans began pushing decisively out of Africa, the stage was already crowded. The genetic evidence suggests that one particularly important wave of Homo sapiens left sometime around 60,000 to 70,000 years ago. Not the first humans to ever set foot beyond Africa. There had been earlier excursions, but the one whose descendants would eventually circle the globe. They didn't walk into an empty world. As they moved along the coasts of the Arabian Peninsula, skirting deserts and following shorelines that offered shellfish, fish, and fresh water, they were entering landscapes that had been Denisovan and Neanderthal territory for hundreds of thousands of years. To the west and north, Neanderthals held their ground. To the east and northeast, Denisovan populations were spread across a vast, patchy range. Mountains, forests, river valleys, perhaps island chains. The map of that time, if we could see it, would show bands of Homo sapiens hugging coasts and river corridors, probing inland where game and climate allowed. Ahead of them, at the edges of the unknown, lived other humans, Not mythological monsters, not faceless cavemen, people. What did that first real contact look like? We will never know the exact day, the exact place. It might have been a narrow valley somewhere in Central or South Asia. It might have been a forest edge where a river emerged onto a floodplain. It might have happened more than once in different regions, centuries apart. But the genetics tell us something simple and astonishing. When our ancestors and the Denisovans met, they did not keep their distance forever. They had children together. So imagine a small group of Homo sapiens moving slowly through unfamiliar terrain. They carry tools that would look familiar all across Africa. Finely made blades, points that could be hafted onto spears, maybe beads or pierced shells worn as ornaments. Their language, whatever it sounded like, allows them to coordinate hunts, describe places, tell stories about where they came from. One morning they see smoke on a distant ridge, not lightning-struck forest, but the thin, careful column of a maintained fire. They're not alone. Up close the strangers are both familiar and strange. Denisovans were probably robust in build, perhaps more like Neanderthals than like us, shaped by lives in harsh climates and rugged terrain. But their eyes would have been intelligent, their hands skilled, their gestures purposeful. These were people who knew their land. They would have had hunting routes, mental maps of water sources, memories of seasons and storms, songs or stories that encoded all that information passed down the generations. The first encounters may have been cautious, watching from a distance, shadowing each other along a river, leaving a carcass untouched at a kill site, noticing later that someone else had butchered it with tools very much like your own. There might have been standoffs, shouts, displays of weapons, mutual retreats. There might have been exchanges, a piece of unusual stone for a skin prepared with a technique you don't quite recognize, a word or two picked up, tested on the tongue, repeated with a laugh when pronounced badly. The DNA pattern suggests that, over time, the caution sometimes softened into something more intimate. When geneticists look at the genomes of people today whose ancestry traces back to Melanesia, Aboriginal Australia, and some Southeast Asian groups, they find a startling fact. Up to 5%, sometimes more, of their DNA clearly comes from Denisovans, not Neanderthals, Denisovans. That much DNA doesn't come from a single brief encounter. It suggests that in some regions the descendants of those African migrants and local Denisovan populations interacted over many generations, long enough for mixed ancestry children to grow up, have their own children, and for those lineages to persist. The most likely region for at least one of these major mixing events is somewhere in Southeast Asia or its gateways, perhaps mainland Southeast Asia, perhaps Sunda, the now submerged landmass that once connected much of Indonesia to the Asian mainland, perhaps nearby island chains. Picture dense tropical forests, river deltas, mangrove coasts, crocodiles in murky waters, birds screaming from the canopy. And somewhere in that vast green maze, two kinds of humans sharing a camp for the night. Maybe a Homo sapiens band following the coast met Denisovans who had already been living there for thousands of years. The Denisovans would know the tides, the poisonous plants, the fish that could be taken easily with spears or traps. Our ancestors might have brought new tool types, new ways of organizing work, new songs. Trade would have been simple at first. Food for stone, hides for ornaments, information for information. There is good water that way. The spirits of this place are dangerous. The rains come earlier in the mountains. But humans have always been more than traders and hunters. They form friendships. They become curious about strangers. They choose partners. Somewhere in those forests, on some beach now drowned by rising seas, a young Denisovan and a young Homo sapiens might have sat on the edge of a firelight circle listening to older relatives talk. They might have shared glances, then words, then a joke. Their parents might have been wary. Their friends might have been fascinated. Feelings don't respect species boundaries, especially not when those species are as closely related as all three post-bottleneck humans were. The genetic distance between a Denisovan and a Homo sapiens was not like the gap between us and chimpanzees. It was closer, softer edged. A child born of such a union would have been fully human in every meaningful sense. 2 arms, 2 legs, a head full of questions. They would have learned whatever language they heard first, whatever stories they were told, whatever skills their parents valued. From the point of view of the village or band, the questions were probably practical, not philosophical. Could this child hunt? Could they gather? Could they help care for siblings? Could they remember where the good waterholes were? As long as the answer was yes, their mixed ancestry would have been an interesting detail, not a metaphysical crisis. Genetic studies hint that this didn't happen just once. There are signs of at least 2 distinct Denisovan lineages contributing DNA to modern humans. One lineage is most strongly represented in Melanesian and Aboriginal Australian populations. Another leaves a smaller, but genetically distinct signal in East Asian populations. That suggests at least 2 major pulses of interbreeding, probably separated by long distances and long stretches of time. One, perhaps in Southeast Asia or nearby, feeding into the ancestors of people who would eventually cross water gaps and settle New Guinea and Australia. Another, perhaps farther north or west in Asia, involving ancestors of some East Asian groups. Imagine those as 2 chapters in a long braided story. In one chapter, coastal and island-hopping bands of Homo sapiens encounter Denisovans who have adapted to tropical forests and archipelagos. Their children carry Denisovan genes that will, among other things, later help with immune responses to local diseases and possibly with metabolizing new foods. In another chapter, farther north or inland, a different set of Homo sapiens groups meets a different Denisovan population, maybe one more tuned to temperate forests or highlands. Here too, some families cross species lines. Here too, genes move. Those Denisovan genes didn't all just sit quietly. Some of them were actively favoured by natural selection. Variants that helped process oxygen efficiently at high altitudes. Variants that helped detoxify certain plants. Variants that tweaked immune systems. All of these could spread through Homo sapiens populations because they were genuinely useful. Behind each of those useful gene variants, though, was a relationship. A conversation by a river, a shared hunting trip, a period of living side by side long enough for people to see beyond they are not us, and into the everyday humanity of the others. It's easy, from a distance of tens of thousands of years, to talk only in terms of admixture events and gene flow. Those are the phrases that appear in scientific papers, but underneath that technical language are lives. People who learned to understand each other's accents, people who argued and reconciled, people who mourned their dead and worried about their children's futures. When you hear that your own genome may contain a small fraction of Denisovan DNA, you're not just hearing about a laboratory result, you're hearing the echo of those meetings. Our species did not expand alone, sweeping a blank map. It moved into a living world of other humans, and relationships formed. Some were hostile, surely. Many were indifferent, but some were tender. Those bonds, fleeting or enduring, left marks that persist in the bodies of their descendants, inside lungs that handle thin air, immune systems that recognize certain threats, metabolisms tuned to particular diets. There are fragments of an ancient, shared story. The Denisovans are gone as a separate people, but the nights when they and our ancestors shared fires, food, and perhaps love are still written in us. The next step in the story is to follow those genetic traces forward, into the present, and see how much of them is still alive in our own biology. When scientists first began to decode those echoes, they expected the Denisovan traces in modern people to be faint, a whisper barely there. After all, this was a population known from a few bones and teeth in a single Siberian cave. How much of them could really have survived? Then the numbers came back. In people whose ancestry traces to Melanesia, to Papua New Guinea, to Aboriginal Australia, the Denisovan signal wasn't a tiny smudge at the edge of the graph. It was bold, persistent. In some individuals, 5, even 6% of their genome is clearly Denisovan in origin. That means if you could line up their DNA like a long scroll, 1 out of every 20 letters, roughly, would be written in an ancient Denisovan hand. This isn't just a genetic footnote. That is the kind of contribution you might expect from a great-grandparent several generations back. Except, in this case, the grandparent is an entire vanished human species, and the Denisovan story doesn't stop there. In many East Asian populations, there's a smaller but still measurable Denisovan component, around 0.2% on average. It sounds tiny, but across billions of people, that fraction represents an enormous amount of Denisovan DNA still moving through human bloodlines. Even in parts of Europe, and surprisingly even in some African populations, careful analysis finds faint Denisovan signals, not because Denisovans themselves marched into Africa, but because some people whose ancestors mixed with Denisovans later migrated back, carrying those fragments with them. Human movement has never been a one-way road, so the Denisovans are not just sleeping in a few isolated island populations. They are, in a very real sense, global. They are present in the genomes of a vast swath of humanity, from tropical coasts to mountain plateaus to crowded modern cities. But those pieces of Denisovan DNA are not all equal. Some segments are scattered randomly, slowly eroding under the churn of generations. Others stand out like bright threads that natural selection refused to let go of. One of those threads runs high into the sky. In the thin, bright air of the Tibetan Plateau, where oxygen levels drop and many lowland visitors struggle for breath, local people live, work, and raise families at altitudes that would leave most of us gasping. Their hearts do not pound as desperately. Their blood does not thicken dangerously with excess hemoglobin. Their bodies handle the altitude as if they were designed for it. In a way, they were. When geneticists looked closely at Tibetan highlanders, one gene in particular stood out, EPA-S1. This gene helps regulate how your body responds to low oxygen, controlling how much hemoglobin you produce and how your blood chemistry adjusts. In most of us, the standard version of EPA-S1 works fine at sea level, but at high altitude it can drive the body into overproduction, pushing blood towards sludge-like thickness. In Tibetans, a distinct version of EPA-S1 keeps that response in check. It's like a finely tuned altitude control system. Where did this special version come from? When researchers compared it to gene variants from around the world, it matched nothing in typical modern human populations. But when they compared it to ancient DNA, the match suddenly appeared in the Denisovan genome. The high-altitude EPA-S1 variant in Tibetans is almost identical to the Denisovan version. Somewhere in the deep past, a Denisovan population that had spent generations in rugged high terrain evolved this adaptation. At some later time, Homo sapiens moving through Asia met them, interbred with them, and carried this high-altitude toolkit away in their DNA. When a subset of those descendants eventually settled on the Tibetan Plateau, the Denisovan EPA-S1 variant gave them a crucial edge. Individuals who carried it were less likely to suffer from altitude sickness, less likely to lose pregnancies, more likely to raise surviving children. Generation after generation, natural selection quietly favoured the Denisovan version, until it became common in Tibetan populations. Today, when a Tibetan child is born with lungs and blood finely tuned to the sky, there is a Denisovan physicist working behind the scenes, invisible but essential. EPAS1 is the most famous Denisovan gene in modern humans, but it is not alone. Other fragments of Denisovan DNA seem to shape our immune systems. In many populations outside Africa, scientists have identified HLA variants, genes that help the body recognize and respond to pathogens, that almost certainly came from Denisovans. Imagine Homo sapiens moving into Asia for the first time. New climates, new plants, new parasites, new viruses and bacteria that their immune systems had never encountered. Local Denisovan populations had already been wrestling with those microscopic enemies for tens or hundreds of thousands of years. Their immune genes carried the memory of those battles. When our ancestors and Denisovans had children together, some of that immunological wisdom was passed on. HLA variants honed by Denisovan struggles became part of the Homo sapiens toolkit. In regions where those variants helped fight off local diseases, they rose in frequency. So when your immune system today recognizes a certain threat and reacts efficiently, there is a small chance it's drawing on instructions first written in Denisovan bodies, long before your own lineage ever walked that land. Other Denisovan contributions are subtler but no less intriguing. There are Denisovan-derived variants that seem to influence how we metabolize fats, tiny biochemical nudges that may once have helped people cope with cold climates, unpredictable food supplies, or diets rich in certain kinds of animal fat or marine resources. Some segments of DNA linked to skin function and adaptation also trace back to Denisovans. These may have tweaked how skin handles ultraviolet radiation, how it maintains its barrier function in dry or cold conditions, how it manages inflammation and repair. Under harsh skies and in rough environments, such fine-tuning can make the difference between thriving and merely surviving. Even teeth bear faint Denisovan signatures. A few gene regions involved in tooth development and morphology show patterns consistent with Denisovan origin. Perhaps in some ancient landscape, a slightly different tooth shape or enamel structure reduced wear from a gritty diet, or made chewing certain tough plants more efficient. The genes that shaped that smile or that bite moved quietly into our lineage and stayed. None of these traits, on their own, tells the whole story, but together they paint a picture of what Denisovan DNA has become in us, a collection of specialized tools forged in other bodies, in other times, that our ancestors borrowed and then never gave back. This is one of the strangest and most beautiful consequences of our closeness to other human species. Denisovans are gone. There is no living band of them somewhere in a hidden valley. Their languages, whatever they were, have fallen silent. Their songs and rituals are lost to memory. No one alive today can say, "My grandmother was Denisovan," in any simple, direct way. And yet, in a different sense, that statement is not entirely wrong. If you are of Melanesian, Papuan, or Aboriginal Australian descent, a significant fraction of your genome is unmistakably Denisovan. If your roots trace into East Asia, the Pacific, or even further afield, small pieces of Denisovan code likely travel in your cells. If you have Tibetan ancestry, every breath you take at high altitude is shaped by a Denisovan inheritance. Billions of people around the world carry at least a few Denisovan sequences. Some of them are probably neutral, meandering along through time. Some have been favored, sharpened by selection, turned into the genomic equivalent of heirloom tools handed down because they still work. The species line that we call Denisovan ended, but its story did not. It branched and flowed into ours. When you think of them now, try not to imagine only a handful of bones in a cold Siberian cave. Imagine instead a living presence diffused through humanity, tiny mosaics of Denisovan ancestry, carried in blood and bone and breath, scattered across islands and continents, hidden in heart cells and brain cells and the delicate linings of lungs. Our history with them was not just about competition for hunting grounds or brief encounters in shadowed forests. It was also about exchange, absorption, transformation. Their DNA stayed. And because it did, some of us breathe more easily on mountaintops, resist certain diseases better, process certain foods more efficiently. Because it did, the human species is not just the story of Homo sapiens alone, but a braided lineage, threads of Neanderthal, threads of Denisovan, woven into the fabric of our supposed modern genome. In a quiet, literal way, the Denisovans are still here, not as an other, but as a part of what we are. The next step is to follow one of those Denisovan gifts in more detail, to climb with it into the highest inhabited places on Earth, and see how a vanished people made possible a life lived in the thin air of the roof of the world. High on the Tibetan plateau, the air feels almost hollow. At 3, 4, even 5,000 meters above sea level, every breath you take contains far less oxygen than your body expects. For most lowlanders, the response is dramatic. The heart hammers, the chest tightens, the brain fogs over. Some people develop headaches, nausea, even life-threatening fluid build-up in the lungs or brain. Stay long enough without acclimatization, and your body begins a frantic biochemical scramble. It pumps out more red blood cells to grab every possible molecule of oxygen. At first, that helps, but there's a cost. Thicker blood flows more slowly. It strains the heart. It increases the risk of clots, strokes, pregnancy complications. What begins as a desperate adaptation can turn into a quiet, creeping danger. Tibetans, on average, do something very different. Their bodies respond to the thin air with remarkable calm. Hemoglobin levels rise only modestly. Their blood stays relatively thin. Their hearts less overworked. Their pregnancies far safer than you would expect at that altitude. Children grow and develop there, not as fragile guests, but as natives. For a long time, nobody knew why. Then a series of discoveries began to connect the people of the sky with the ghosts in our genome. In 2019, on the remote, wind-scoured Tibetan plateau, researchers took a closer look at a fossil that had actually been found years earlier by a local monk. It was a mandible, a thick, heavy lower jaw, discovered in a cave at Xiahi, more than 3,000 meters above sea level. It didn't look like the jaw of a modern human. It lacked the chin we take for granted. The teeth were broad and sturdy. When scientists tried to extract DNA, the genetic material was too degraded by time and altitude. But there was another approach. They turned to ancient proteins preserved in the fossil, molecular fragments that can still carry an evolutionary signature. The result was astonishing. The Xiahi jawbone did not match Homo sapiens. It did not match Neanderthals. Its closest known relatives, based on those surviving proteins, were the Denisovans. In other words, at least 160,000 years ago, long before Homo sapiens had spread widely into Asia, a population of Denisovans was living at extreme altitude on the Tibetan plateau. This wasn't a brief excursion. To leave a fossil there, to be part of a local ecosystem, they would have needed to survive the cold, the wind, the thin air for generations. Long enough for natural selection to test every weakness and reward every small advantage. Somewhere in that long experiment with life in the sky, a particular version of a gene called EPAS1 began to spread. EPAS1 is part of your body's oxygen sensing system. When oxygen levels drop, EPAS1 helps trigger a cascade of responses, upregulating genes that increase red blood cell production, altering blood vessel growth, fine-tuning how tissues use the oxygen they get. In most lowlanders, this system was honed for life at or near sea level. Take that machinery and put it on a mountaintop, and it overshoots. It keeps pushing, keeps thickening the blood, because from its point of view the emergency never ends. The Denisovan-like version of EPAS1 that Tibetans carry works differently. It seems to blunt that runaway response. It allows a moderate adjustment to low oxygen without tipping into dangerous overproduction of red blood cells. It's like having a thermostat that doesn't immediately crank the heat to maximum the moment the room gets chilly, but instead responds in a measured, controlled way. When geneticists compared EPAS1 in Tibetans to the same gene in nearby lowland populations, the contrast was stark. Among Tibetans, this special variant is extraordinarily common, present in the vast majority of people. In closely related Han Chinese populations, it is almost absent. That pattern is a signature of powerful natural selection. If a random mutation arises that doesn't do much, it drifts around at low frequency, sometimes rising, sometimes falling, its fate largely a matter of chance. But if a variant appears that confers a huge advantage in a particular environment, protection from disease, resistance to cold, the ability to bear healthy children at high altitude, its frequency doesn't just drift, it rockets. In the case of EPAS1, the rocket's trail leads straight back into deep time. The Tibetan high-altitude variant is extremely rare worldwide. It doesn't look like the typical version seen in other modern human populations. But when researchers lined it up against Denisovan DNA, the resemblance was almost perfect, not a vague similarity, a near match. The simplest explanation is also the most profound. This EPAS1 variant did not originate in Homo sapiens at all. It evolved in Denisovans, probably in populations like those that left the Siahi jawbone in that cave above the clouds. Later, when Homo sapiens expanded into Asia and met Denisovans, some of their children inherited this high-altitude toolkit. For most of those children, living at lower elevations, the Denisovan EPAS1 variant was probably just another neutral passenger in the genome, neither especially helpful nor harmful. But when some of their descendants moved back up onto the Tibetan plateau, everything changed. Suddenly, having the Denisovan version became the difference between chronic strain and relative ease, between repeated pregnancy loss and viable births, between a lineage that slowly dwindled and one that flourished. Over thousands of years, generation after generation, natural selection quietly amplified that borrowed Denisovan gene until, in modern Tibetans, it became almost universal. EPAS1 is the clearest case we know of, but it may not be the only Denisovan gift to people living in extreme environments. Genome studies have identified other regions in Tibetan and neighbouring highland populations that show signs of adaptation to altitude, variants that affect nitric oxide pathways, blood vessel function, iron metabolism, even lung development. Some of these regions also bear hints of archaic origin, with patterns that don't quite fit typical modern human diversity. The picture is still blurry. Ancient DNA is difficult to obtain from many parts of Asia. Denisovan fossils are rare, but as more genomes are sequenced, more subtle echoes are emerging. Possible Denisovan contributions to high-altitude adaptations in populations across the Himalayas, and even in parts of the Andes, where some signals may represent parallel solutions, others borrowed ones. It's too early to say how many of these adaptations are truly Denisovan in origin. The science is unfolding in real time. But the EPAS1 story offers a template. Ancient humans moving into harsh landscapes, encountering local cousins who have already weathered those conditions for ages, and leaving with some of their solutions encoded directly into their DNA. This reframes what interbreeding meant. It's tempting to picture ancient humans as rival species locked in a zero-sum struggle. Us versus them. Homo sapiens versus Neanderthals versus Denisovans, each guarding their territory, their food, their mates. There was certainly competition, but there was also this: exchange, blending, a shared genetic marketplace where useful traits could move from one lineage to another. Far from being a weakness, that permeability may have been one of our greatest strengths. Homo sapiens did not have to reinvent every adaptation from scratch. When our ancestors wandered into high plateaus or strange forests, or disease-ridden river valleys, they sometimes met people who had already paid the evolutionary price of living there. Their bodies carried hard-won solutions, immune defenses, metabolic tweaks, altitude hacks. Through interbreeding, we could, in a sense, license those solutions, take them on, fold them into our own evolving genome. Denisovans seem to have been especially good at surviving in extremes. From Siberia's freezing river valleys to the roof of the world in Tibet, to the rugged islands of Southeast Asia where sea crossings were required, their genetic fingerprints show up again and again in places where life is not simple. The Xiahi jawbone is just one fossil, lying silent in its mountain cave. The EPAS1 gene is just one stretch of code, humming quietly inside human cells. But together they hint at a broader truth. Denisovans were not a marginal, fragile people clinging to a single refuge. They were adaptable, widespread, capable of thriving in environments that would test any human body. And because our ancestors met them and had children with them, some of that hardiness did not vanish when Denisovans did. It passed into us. When a Tibetan herder walks a high ridge without gasping for air, when a child in Lhasa runs and laughs under a cobalt sky that would dizzy most of the world, when hearts beat steadily in a place where oxygen is scarce, there is an ancient partnership at work. Not just Homo sapiens, but a quiet Denisovan inheritance. A reminder that human success has never been the story of a single, isolated species, but of a family of humans, overlapping in time and space, sharing not only lands and tools, but genes. High above the clouds, in every breath that comes easily, the Denisovans survive. But if their genes helped us, why did they disappear? How can a people so adaptable, so widespread, leave behind only fragments of bone and a scattering of sequences in our cells? To answer that, we have to follow their trail not into the sky, but into the long twilight of their existence, the slow vanishing of a human species whose body is gone, but whose legacy refused to die. The first thing to understand is that the Denisovans did not vanish in a world that was calm and stable. They lived in the late Pleistocene, a time when climate did not simply drift, it lurched. Ice sheets advanced and retreated like a slow, grinding tide. Forests became grasslands, then forests again. Coastlines shifted dozens of kilometers as sea levels rose and fell. Rivers changed course. Entire ecosystems appeared and disappeared within the span of a few thousand years. For a species spread from the frozen river valleys of Siberia to the humid islands of Southeast Asia, that instability was both an opportunity and a threat. When the ice advanced, northern habitats shrank or vanished. Herd animals migrated south. New corridors opened, old ones closed. When the ice retreated, new lands were exposed, but familiar hunting grounds flooded under rising seas. A population could thrive for thousands of years along a rich coastal shelf, only to see that shelf swallowed by the ocean, its people forced inland into already occupied territories. Denisovans had proven they could adapt. High altitude, deep cold, tropical forests. They met all of these and survived. But adaptation takes time, and time was exactly what the late Pleistocene climate kept taking away. Every rapid shift would have hit them like a slow-motion disaster. Not an asteroid in the sky, but 100 small cuts. A key prey species moving out of range, a valley drying up, a migration route blocked by advancing glaciers, a low-lying homeland drowned as sea levels climbed at the end of an ice age. In isolation, any one of these might be manageable. Together, layered over tens of thousands of years, they turned survival into a constant balancing act. and then into this churning, unstable world walked us. Homo sapiens had been evolving in Africa under the same broad climate pressures, but with a different set of responses. By the time small bands of our species began spilling into the Middle East, then deeper into Eurasia, around 60,000 years ago, they carried with them a strange new toolkit. Not just stone blades or fire, but something harder to see in the fossil record. Dense social networks, rapid cultural innovation, and the ability to move information across space and time with extraordinary speed. Where Denisovans and Neanderthals seem to have lived in relatively small scattered groups, Homo sapiens gradually stitched those groups together. Long-distance trade networks in obsidian, shells, pigments, shared styles of tools and ornaments across vast regions. Evidence that ideas could travel farther than any single person ever walked. That kind of connectivity is an evolutionary force in its own right. If a Denisovan group somewhere perfected a new hunting technique, it might spread slowly, radiating through neighbouring bands over generations, if at all. If a Homo sapiens group did the same, the idea could jump hundreds of kilometres in a few lifetimes, carried by marriage ties, exchange partners, visiting allies. In a world where the environment wouldn't sit still, the winners were not necessarily the strongest, or even the smartest in some absolute sense. They were the ones who could redesign their way of living the fastest. Homo sapiens seemed to have been particularly good at that. So when our ancestors moved into Asia, they weren't just another cousin on the landscape. They were a moving front of populations that could recover quickly from losses, regroup, and adapt their strategies on the fly. Larger networks meant more mates, which meant more genetic mixing, which reduced inbreeding and buffered against local disasters. More minds in contact meant more experiments, more innovations, more chances for something useful to catch on. For Denisovans, encountering Homo sapiens may have felt at first like just another meeting with strange neighbours. There were probably exchanges of goods, of techniques, and of genes. Some Denisovan groups likely benefited from contacts with these new arrivals, picking up tools or strategies that worked. But over thousands of years the balance began to tilt. Imagine a patchwork of territories across Asia. In some regions, Denisovans are numerous, their presence deep, their adaptations finely tuned to local conditions. In others, they are sparser, their settlements thin, their hold on the land more tenuous. Now imagine Homo sapiens groups arriving at the margins of this patchwork. They settle in places where Denisovan presence is weak, or where recent climate shifts have created openings, abandoned valleys, newly exposed coasts, river corridors following retreating ice. They expand, join with neighbouring bands, grow in number. In areas where both species meet, there is interbreeding, some conflict, some cooperation. Over time, two different processes unfold together. The first is classic competition. In certain environments, Homo sapiens may simply have been better at squeezing more food, more children, more allies out of the same landscape, more efficient hunting strategies, more flexible diets, more complex division of labour between individuals. In those places, Denisovan groups could slowly lose ground, not catastrophically, not all at once, but through a gradual shrinking of their territories, a decline in group size, a reduction in the number of safe places their children could grow up. The second process is quieter, and in some ways more profound. Absorption. Genetics gives us an unexpected hint. Modern populations in Asia, Oceania, and parts of the Americas don't carry trace amounts of Denisovan DNA in just one pulse, as if there had been a single brief episode of mixing. Instead, the patterns suggest multiple episodes, scattered through time and space. Different Denisovan-like segments in different populations, some older, some younger. That looks less like a single collision, and more like a long, complicated relationship. Every time a small Homo sapiens group met a small Denisovan group and had children, some of those children may have grown up in predominantly Denisovan communities, others in predominantly sapiens ones. In the next generation, some of them may have moved, married out, or been traded or adopted into other groups. Their descendants would have carried Denisovan ancestry into communities where Denisovans themselves were rare or absent. Repeat this, not for a century, but for tens of thousands of years. Slowly, the genetic boundary between the two species blurs. In some regions, Denisovans may have remained distinct for a very long time, maintaining their own traditions, their own identities, even as they mixed at the edges. In others, especially where their numbers were small, those distinct communities may have melted into the expanding Homo sapiens networks. From a genetic standpoint, this is not extinction in the way we think of mammoths or saber-toothed cats. It is more like a drop of ink diffusing through water. The form dissolves, but the molecules remain, scattered, diluted, yet still very much real. This is the absorption hypothesis. The Denisovans did not necessarily vanish in a single dramatic collapse, but faded as a separate visible population while their DNA flowed into ours. Neanderthals, on the other side of Eurasia, tell a strangely similar story. They too disappear from the fossil record around 40,000 to 30,000 years ago. They too leave behind fragments of their DNA in modern non-African populations. The same broad suspects lurk in the background: climate instability, competition with Homo sapiens, repeated small-scale interbreeding that gradually transferred their genes into our lineage, even as their last distinct bands dwindled. Two sister species separated by half a world, fading from sight in roughly the same window of time under similar pressures. Was this just coincidence? Or does it hint at a broader pattern? A moment in history when Homo sapiens, once a small branch on the human family tree, became the trunk. The honest answer is that we don't know everything we would like to know. Denisovan fossils are still astonishingly rare. Most of what we infer comes from genomes, from a handful of sites, from indirect evidence woven together into careful but still fragile stories. New discoveries could shift the picture. A single well-preserved skeleton in Southeast Asia, a new cave in the Himalayas, another mandible on some forgotten plateau, and we might have to redraw entire maps of their final millennia. It is possible that in some region we have not yet sampled, Denisovans held on far longer than we think. It is possible that they suffered localized catastrophes we have no trace of yet. A volcanic eruption, a disease swept along trade routes, a chain of unlucky winters. It is possible that their cultures were as rich and varied as our own, but built mostly of wood, fibre and story, all things that rot or blow away. Sitting with that uncertainty can feel uncomfortable. We are used to neat endings: rise, fall, conclusion. Extinction events with clear causes, a meteor, a plague, a date on a timeline that says, here, this is where they ended. Denisovans resist that kind of closure. What we can say, with some confidence, is that around 30,000 years ago, the world's last clearly Denisovan communities were probably gone. No more children who would have recognized themselves as part of that lineage. No more elders telling their particular histories around particular fires. And yet, on some level, they remained. Not as bones in a cave, but as sequences of nucleotides winding quietly through the genomes of people from Siberia to New Guinea, from the Andes to the Himalayas. In blood that handles oxygen a little differently. In immune systems that shrug off certain pathogens a little more effectively. In subtle shapes of faces and skulls that we do not yet know how to read. An entire species can vanish in body yet persist in code. When you lie awake at night and trace your own breathing, your own heartbeat, it is worth remembering that part of what keeps you alive was once tested in other bodies, in other minds, under other skies. Denisovans did not leave us a written language or standing monuments of stone. What they left is more intimate and more enduring. They left themselves folded into us. In the end, wondering why they disappeared is also a way of asking a deeper question. What does it mean for a human species to exist, or to end, when the boundaries between us have always been more porous than we imagined? To explore that, we have to leave bones and genes behind for a moment and listen for echoes of Denisovans in a different realm altogether, not in the ground, not in the blood, but in stories. If Denisovans left themselves folded into our DNA, it is natural to wonder, did they also leave traces somewhere else? Not in stone tools or hearths, but in the long, fragile medium of human memory. Not the kind of memory that shows up in documents or inscriptions. Something older and more slippery, the kind that travels by voice, by campfire, by lullaby. The kind that changes a little with every telling, but somehow never quite disappears. Anthropologists call it oral tradition, myth, folklore. Now, we have to be careful here. Stories are not fossils. You can't carbon date a legend or sequence the genome of a myth. But when you realize that for tens of thousands of years our ancestors shared the world with other kinds of humans, it becomes difficult not to look at certain stories and feel a little prickle at the back of the neck. Across the high plateaus of Tibet and the rugged ranges of the Himalaya, people tell tales of wild folk in the mountains, hairy, powerful, somewhere between human and animal. They're not exactly monsters, not exactly spirits, not exactly people. Sometimes they are dangerous, sometimes just elusive. They live where humans do not quite belong, in thin air, deep snow, places where survival feels like a dare. The Yeti, the Megoi, the wild men of the snows. Far to the north, across Mongolia and Siberia, there are stories of forest beings and mountain people, solitary or in small bands, big, strong, shy. Sometimes they steal food, sometimes they steal brides or children, sometimes they teach secrets, like where to find water or how to survive a bad winter. In the jungles of Southeast Asia, the pattern repeats. Forest spirits, little people, hairy men who live just beyond the last fields, who are clever with traps and vanish when you try to follow them. They're often described as almost human, not quite us, but not entirely other. Are these memories of Denisovans? Probably not in any direct literal sense. The gap is too long. We're talking about encounters that, if they happened at all, happened tens of thousands of years ago. Oral traditions are powerful, but they are not tape recorders. They change as cultures change, as languages shift, as religions rise and fall. More likely, these figures are what humans always create when faced with wilderness. We populate the edges of our maps with beings that embody our fears and desires. The wild man, the forest woman, the mountain giant. They stand in for everything that is not domesticated, not fully under our control. And yet, there is a strange resonance when you place these stories against the genetic map. In many of the same regions where tales of wild mountain or forest people circulate, Tibet, the Himalayas, parts of mainland and island Southeast Asia, modern populations carry some of the highest known levels of Denisovan ancestry. The people who tell the stories are, in a quiet genetic sense, the descendants of those others. Coincidence? Almost certainly at the literal level, but at a symbolic level it is hard not to feel that something is happening here. A kind of unconscious recognition that our past was not solitary, that there were once other eyes in the dark, other hands shaping the same landscapes, that the boundary between us and not us has never been as clear as we pretend. On one small island in Indonesia, the speculation sharpens. The island is Flores. The species is Homo floresiensis, the so-called hobbit, with its tiny body and small brain, yet strangely advanced stone tools. They lived there until perhaps 50,000 years ago, maybe even more recently, depending on how you interpret the evidence. The people of Flores have a story about small hairy folk who used to live in the deep forests. They called them the Ebugogo. The Ebugogo, according to local accounts collected in recent centuries, were about 1 metre tall. They walked upright. They could speak, but their language was strange, more like murmuring or chattering than proper speech. They stole food, especially from gardens. They were clever at surviving, but not very bright in other ways, and they were said to have been present not in some mythic time before time, but as recently as a few hundred years ago, before being driven into hiding or wiped out. When archaeologists first uncovered Homo floresiensis, these tales of the Ebugogo took on a new, eerie flavour. Here was a small-bodied human species, actually known from the ground beneath the island, whose last fossils were not impossibly ancient. Here were detailed stories, preserved by local people, about small-bodied, almost human forest dwellers on the same island. Is this proof that oral traditions can preserve the memory of another human species? No. The connection is tempting but contested. The fossils and the folk tales do not line up cleanly enough in time to satisfy everyone. Many scholars point out that stories of little forest people exist in many cultures that never lived alongside another human species at all. But even if the Ebugogo are not a direct memory of Homo floresiensis, they remind us of something important. The human mind is very willing to imagine neighbours that are almost, but not quite like us. And for much of our deep history, those neighbours were not imaginary. Denisovans, Neanderthals, hobbits on Flores. Our lineage didn't climb a ladder alone. It grew in a thicket tangled with others. We touched, traded, fought, loved, and had children across species lines. The myths of wild people at the edge of the world may not be photographs of those encounters, but they feel like an echo of the emotional landscape they created. For a long time, many scientists and philosophers preferred a different story. Homo sapiens as a pure, separate lineage, a species that arose, perfected itself, and then simply replaced its cousins. Clean lines, clear categories. Denisovan DNA in modern humans has shattered that simplicity. We're not a pure species, we are a mosaic, pieces of different human lineages, layered and interwoven. Neanderthal fragments helping to shape aspects of our skin and immune systems. Denisovan segments tuning our blood to thin air or shifting how we respond to certain pathogens. Possibly other, still unidentified archaic contributions lurking between the known bits. This realization carries a quiet philosophical weight. When you look at a crowd of people, you're not looking at the triumphant survivors of a single unbroken line. You're looking at the living archive of an entire clade of humans, most of whose distinct branches have vanished as cultures but not as code. What does that mean for how we think about us and them? If our own bodies are already mixtures, if modern human is a blend of past species, then the idea of clean separation starts to feel thin. The borders we draw between groups, between populations, between kinds of people, are revealed for what they are, recent, superficial, laid over something much older and more entangled. For some, learning they carry Denisovan DNA is just an interesting footnote on an ancestry report. For others, it lands more deeply. It invites a sense of continuity with vanished worlds. The knowledge that your ability to stand on a Himalayan pass, or walk through a Melanesian jungle, or fight off a particular virus, was partly shaped by people who would not have called themselves sapiens at all. Today, consumer genetic tests sometimes include an estimate of archaic ancestry. This percentage Neanderthal, that percentage Denisovan, if your background ties into the right regions. The numbers are usually small, 2%, 3%, maybe a bit more in parts of Oceania. But those percentages represent thousands of tiny segments of DNA, each one a surviving thread from encounters in Pleistocene caves and river valleys, each one passed down, parent to child, for hundreds of generations, through wars and migrations and quiet domestic lives, until they ended up in you. You can sit in a brightly lit room, surrounded by devices and structures no Denisovan could have imagined, and still be carried, in part, by their legacy. Some people, seeing their Denisovan fraction on a screen, feel a kind of ancestral curiosity. Who were they? What did they see? What did the world smell like to them, high on a cold plateau or deep in a tropical valley? Others feel an odd tenderness or grief for a species that is gone in one sense but still alive in another. Does knowing that extinct humans live on in us create obligations? Perhaps it suggests a different kind of humility, a recognition that we are not the culmination of a straight line, but the current expression of a long branching experiment, that our minds and bodies are collaborative works, built by countless lives from countless lineages. It might change how we think about extinction itself. If a species can disappear culturally while surviving genetically, what exactly are we losing when a branch of the tree falls? And what might we be erasing unknowingly as our own species continues to reshape the planet? These are large questions, and they do not have simple answers. But they all flow from the same surprising source, a few fragments of bone in a Siberian cave, and the ancient code extracted from them. The more we learn about Denisovans, the more they press us to reimagine what it means to be human, not as a solitary figure on a pedestal, but as a chorus, a blend of voices, some of which we can no longer hear directly. To listen more clearly, we have to keep searching, not just in myths and memories, but in caves, in sediments, in the molecules of our own cells. What comes next is not just more digging, it is learning to read traces so faint that, until recently, no one even knew they were there. For a long time, the rule in paleoanthropology was simple: no bones, no story. You needed a skull, a tooth, a fragment of jaw, something you could hold, measure, name. Denisovans began to undermine that rule from the start. A fingertip, a tooth, almost nothing. And yet from that almost nothing, an entire hidden population emerged. Now the tools are changing again. In the last few years, scientists have learned to look not just at the bones in a cave, but at the dirt between them, the dust on the floor, the fine sediments that packed into cracks in the rock. Inside those grains of dust are microscopic fragments of DNA, molecules shed in flakes of skin, in droplets of blood, in the decay of feces and urine. Long after bones have crumbled away, these fragments can linger, clinging to mineral surfaces, protected from sunlight and microbes. This is called environmental DNA, eDNA. In places like Denisova Cave, researchers now take spoonfuls of sediment from different layers and extract whatever genetic material they can find. They do not know in advance what will appear. It might be wolf, cave bear, bison, hyena, or it might be human. In some layers with no visible human bones at all, eDNA has revealed a quiet presence, Neanderthal, Denisovan, sometimes both. It is as if the cave itself has become a kind of time-resolved census, capturing which species passed through over tens of thousands of years. The implications are enormous. There are caves across Eurasia with tantalizing stone tools, hearths and cut-marked animal bones, but no clear human fossils. For decades, arguments have raged. Were these made by Neanderthals, by early Homo sapiens, by some other group? Now a few grams of sediment can tip the scales. Extract the DNA, sift through the reeds, and you may find Denisovan sequences woven into the genetic noise. Not a bone, not a skull, but a molecular ghost saying, "We were here." Already, eDNA has extended the known range of Denisovans to caves where they were never suspected. Each new signal redraws the map, stretching their presence farther across Asia into time periods that did not seem to belong to them before. If Denisovans can be found in dust, then any undisturbed cave or rock shelter anywhere from Siberia to Indonesia becomes a potential archive, a place where absence of bones no longer means absence of people. But even so, bones matter. So far, almost all our confirmed Denisovan fossils come from one place, Denisova Cave in the Altai Mountains, a tooth here, a jaw fragment there. Enough to anchor a genome, but not enough to show what an entire body looked like, or how much variation there was between Denisovan populations. Genetics tells us very clearly that Denisovans must have lived far beyond this single Siberian cave. Their DNA is strongest today in people from New Guinea, parts of the Philippines, and island and mainland Southeast Asia. That pattern does not happen by accident. If modern populations in Oceania carry up to 5% Denisovan ancestry, then long ago real Denisovan groups must have lived, hunted, cooked, and slept somewhere in that region. They must have left bones in caves, teeth in river sediments, footprints in mud that turned to stone. We just have not found them yet. Or, more unsettling, we have found them, but have not recognized them for what they are. There are scattered fossils from China, Indonesia, and other parts of Asia that do not fit neatly into Neanderthal or modern human categories. For years, they were given local names, debated, filed under archaic, and left in museum drawers. Now, researchers are going back to these ambiguous bones with new techniques. Sometimes there is no extractable DNA left. The climate is too warm, the fossils too old, the molecules too broken. Ancient DNA decays like an unwound thread. But proteins survive longer. The hard tissues in bones and teeth are threaded with durable proteins, especially collagen. Their amino acid sequences differ slightly from species to species. You can think of them as another kind of code, a rougher, more durable cousin of DNA. Paleoproteomics, the study of ancient proteins, allows scientists to grind a tiny sample from a tooth or bone, run it through a mass spectrometer, and reconstruct these sequences. Even when DNA has faded past recovery, proteins can sometimes still whisper a name. Already this approach has been used to identify Neanderthal material where DNA was gone, and to suggest Denisovan affinities for certain Asian fossils. In time, it may reveal Denisovans in places where no one thought to look, hidden in collections gathered decades ago. A jaw from a cave in China, a tooth from a limestone hill in Laos, a fragment from a riverbank in Borneo. Each one, when tested, might shift from mysterious archaic human to confirmed Denisovan. And even as we chase Denisovans in the rocks and dust, another frontier is unfolding in the molecules inside living people. The first Denisovan genome was astonishing, but it was only the beginning. As more modern genomes are sequenced from across the world, especially from underrepresented regions, strange patterns keep surfacing, segments of DNA that look ancient but do not match Neanderthals or Denisovans. These are sometimes called ghost lineages. They represent archaic human populations that must have existed, interbred, and left descendants, but whose bones, so far, we have not identified, or whose DNA we have not yet sequenced from fossils. Some of these ghost signals show up in African populations, where the fossil record is rich but the ancient DNA record is sparse. Others appear in parts of Asia, nested inside Denisovan-like sequences, yet distinct from the Denisovan genomes we know. It's possible that what we call Denisovans were not a single uniform group, but an entire cluster of related populations spread across a continent, mixing and separating over hundreds of thousands of years. Some branches may have interbred more with Neanderthals, others with early Homo sapiens, others with still unknown archaic neighbours. From our vantage point today, with just a few ancient genomes in hand, these lineages blur together into something we label Denisovan. As more data arrives, that blur may resolve into multiple named populations or even new species. The human family tree, once drawn as a simple trunk and a few side branches, now looks more like dense undergrowth, twisting, recombining, looping back on itself. Somewhere in that tangle, Denisovan ancestry threads through like a persistent vine, crossing from branch to branch, and all the while, caves wait. Across the mountains of Central Asia, the karst landscapes of southern China, the limestone islands of Southeast Asia, and the volcanic ridges of Indonesia, there are thousands of caves that have never been systematically explored. Many are hard to reach or politically unstable